the record. All righty, excellent. Today is Thursday. It's the end of our week, February 25th, 2021, of course. And we are going to start the same thing we're going to be doing for the next couple weeks. In lecture, we are going to be talking about the physiology of the bones and the skeletal system. And then in lab, you guys will be presenting your bones and bone features. Today and Tuesday, we'll do the axial skeleton. Also on Tuesday, you'll be assigned your appendicular bones, which we'll do Thursday and the following Tuesday. You have 4 assignments, Unit 7, 8, and 9, that are same as they ever was on those. And then again, that 30-point skeletal review, which I believe becomes available today. I don't remember exactly when I made that available. It might not be till next week, because like I said, I don't want you working on it yet. But again, it is going to be created for correctness about putting the pieces of the bones together and the overall shape of the skeleton, all leading up to Thursday the 11th, when we have our lab and lecture exam. All right, so any questions on that before we get started? Oh, that's the other thing I need to test. Exams. I forgot to look up the scores, but let's say that they were not pretty. Hold on. Yeah, totally. Yeah, again, for your presentation, that would totally be fine. You know, to have a picture with the number pointing at it and saying that's there or something like that. That's totally fine. Absolutely. The exams, again, very wide range of scores. And I kind of warned you that this was going to be the case. So we talked about this a little bit in the last class as well, how this last exam with all of the histology stuff tends to be challenging for a lot of people that don't have a lot of microscope experience. And that definitely showed on the lab exams. Lecture exam was better, but definitely could be better. So again, after class today, I will be releasing the exams for 24 hours so that you have access to them. And then on Friday afternoon, I will close them down again. Half today and half Tuesday. So like I said, my goal today for the first day is going to be to just get through the skull. So that's going to be my hope for today. And then next week, we'll do the rest. Yes, the lab and lecture exam place that are there in the grade book right now for exam 3 are just placeholders, so that as you're looking at the schedule, you're looking at the assignments, you're looking at those things, you can see the day that it is going to be on. But I haven't finalized the exams yet. And because the lab exam varies from semester to semester of how many questions on it, I don't have a set exam for yet. So it's there as a placeholder, that's all. Well, it'll be available after class today. So if you're traveling tomorrow, then you'll have plenty of time today to look at it after class. Usually, like I said, I like to have it available for about 24 hours. So I guess we get done with 4. So probably somewhere around 4 tomorrow will be when I close it down as well. And again, remember, if you don't have time to see it during those times, you're always welcome to come to my office hours, and we can review it and go over it together that way as well. All right. Any other questions on that? The good news is, like I said, your grades are based solely on, not solely on the exams, but on every point in the class. And so we're still at about a 70%, but those definitely need to come up. And the good news is from here forward, it does get easier. And again, it also should be getting easier because you should get better at anticipating the types of questions that I'm going to ask you based on the information we cover. And that's actually a perfect lead in to what we're going to be talking about next. We are going to be talking about the processes, the physiological processes involving bones and maintaining bone homeostasis. For starters, we start as a big bundle of cells inside our mommy's tummy. And from there, we develop bones. And there are 2 ways to make bones. We're going to learn both ways. And when there are 2 ways to make bones, or really 2 related processes like that, how many possible essay questions can there be?
2.
Okay, the 2. Can we do any more? We could describe process 1, describe process 2, but is there anything else that we can do with related processes?
Compare and contrast.
Compare them, absolutely. So there's right off the bat 3 possible essay questions off of the stuff that we're going to talk about today for developing bones. We are going to talk about once you make a bone, we need to grow that bone, both in width and in length. So we got two related processes there. Of course, once we get to be grown up, big people, adults like we are now, our bones may not be getting bigger and wider, but we still need to be able to maintain those bones. And so we're going to talk about the three important characteristics necessary for maintaining bones. And occasionally bones will break. And when they break, we have to talk about how we're going to repair that damaged bone. So we're going to talk about how you break them and how you repair them as well. The other thing that we hinted at in the last class, and I want to definitely emphasize again as we go through this material, is the other important thing that's going on with our bones is the homeostasis of calcium. Right? We have two major issues here. Calcium makes cells do wonky things, and we absolutely positively want cells to do wonky things. However, 99% of our calcium is embedded in our bones. So as we talked about in the last class, if it comes down to a choice between having the right amount of right amount of calcium in our blood or having really, really dense bones, who's going to win every time? Is it more important to have the right amount of calcium in the blood, even if it means taking it out of the bones and weakening the bones? Or is it more important to have strong bones and screw everything else in the system that needs calcium?
The blood.
The calcium.
blood.
Yeah, blood calcium, absolutely. So while we're going to be talking about maintaining homeostasis of the bone, big, strong, healthy bones, we have to remember that calcium homeostasis is even more important than that bone homeostasis. And so we'll talk about how those two are related to each other. All right, so again, we've got a very fun path ahead of us here of stuff to talk about in lecture. And then, like I said, we get to finally switch gears in lab and do more labby type stuff. But let's start here with bone formation. Again, as I mentioned, we start with a bundle of cells. So again, the people doing the skulls are the people presenting today. We talked about that at the beginning, but none of that is till after lecture. So let's not worry about that till then. Big bundle of cells in mommy's tummy, and our starting embryonic skeleton is primarily made up of two types of components. One is a fibrous connective tissue, and the other is hyaline cartilage. And we happen to have two ways of making bones and two starting points in tissues, so that should not be a surprise there. Of course, the process of making bone is the process of osteogenesis. Osteogenesis or ossification starts very early in the embryonic development at about week 6 or 7 of that embryo's development inside of mommy's tummy. And with two different tissues and two different starting points, we have two different bone formation processes. The first is endochondral. And again, anytime we have these alphabet soup terms that you know you're going to have to spell correctly at some point, you break them down into the parts. Endo, of course, means within. Chondrial refers to cartilage. So endochondral ossification is making bone within hyaline cartilage. Intramembranous, intra means, well, within. Membranous, within a membrane. Intramembranous ossification is making a bone within a fibrous connective tissue. So let's talk about endochondral ossification first. This is the more common type. This is what is used for most of the bones of the body, and in particular, all of the long bones. And that is really tiny. All of the long bones are made using endochondral ossification. We spent a lot of time talking about the anatomy of a long bone, so it's probably important to talk about that and look at that way. Of course, before we can talk about endochondral ossification, so before we have to remember that we are making this bone from a cartilage model of the bone. And so the first thing we have to do is we have to make our cartilage model. Remember when we talked about tissue healing in the integumentary system. And again, this is something that a couple of people got confused about on the lecture exam. We talked about regeneration and fibrosis as those two healing processes. And we talked about how those two healing processes are basically used by all the tissues of the body, right? Depending on the severity of the injury, depending on the type of tissue that's injured, we are going to either use regeneration or fibrosis for all the tissues in our body. Now, the examples we used happen to be in relation to the skin with a superficial and a deep a type of wound, but these are things that we're going to use in other organ systems when we talk about the healing of them as well. And it's the same thing here. We are going to talk about using a chunk of cartilage that is shaped like a bone to make a bone out of bone connective tissue. However, to do that, we have to grow our cartilage model first. And there are two main growth processes we are going to use. And do you think these growth processes are just used in cartilage? Well, the fact that I'm sitting here waving my hands so much with so much emphasis about it should be a strong indication that no, we're going to use these growth processes for other things as well. So we need to talk about these growth processes. Of course, cartilage has to come from cells. Those chondroblasts are going to come from those pluripotent stem cells known as mesenchymal cells. And again, to beat the dead horse, these are indeed pluripotent stem cells. And what does that mean again?
They can produce a small range of different.
Yeah, exactly. They can produce a small range of cells. And in particular, mesenchymal cells produce all of the cells associated with connective tissues. So not just chondroblasts, but osteoblasts and adipocytes and fibroblasts and everything else in between. So here we have a chunk of cartilage. That chunk of cartilage, even though this illustration doesn't do the best job of showing it, we know is going to have a thin layer of dense irregular connective tissue wrapped around its outer surface. And as we know, it is going to house within it some of those mesenchymal stem cells that are going to be able to divide to produce new chondroblasts. And so our goal is to start from a small, tiny chunk of cartilage and get to this shape. I'm not sure what you mean by transported. Mesenchymal cells are stem cells. They're pluripotent stem cells found in all sorts of different places, like for instance here underneath the perichondrium, that are able to divide to produce, and a mesenchymal cell, which are found in lots of places, can divide to become any type of connective tissue you need. So when we need to make bone, we can start from mesenchymal cells. We need to make cartilage, we can start with mesenchymal cells. Adipose tissue, mesenchymal cells, and so on and so forth. No, the ones that people extract from the umbilical cord, remember, are the omnipotent stem cells. Omnipotent stem cells are the ones that can divide to become anything. And so those are the ones that come from embryos. Those are the ones that come from fetuses. Those are the ones that come from the umbilical cord blood and things along those lines. Unless you are pregnant, you do not have any omnipotent stem cells in your body at this stage in your life. But you have plenty of pluripotent and unipotent stem cells. So everybody here has a ton of stem cells in their body. They're just unipotent and pluripotent. The omnipotent ones are the ones that come from the babies, and they can become anything. All right. So let's do this. Actually, I want to draw mine first. Whoops, no, didn't mean to do that. So let's start with a chunk of cartilage. Here's a chunk of cartilage. And as we just finished talking about, that chunk of cartilage is going to have a perichondrium on its outer surface. And as we just finished talking about, it is going to house some mesenchymal cells. But because we know this cartilage is a solid matrix, we know that there is also going to be, nope, don't want that to be green, a space in the center of the matrix. And what would the space in the center of this matrix be?
Lacuna.
A lacuna, excellent. And within this lacuna, we are going to have a chondrocyte. All right. So again, this is what a small chunk of cartilage looks like. Our goal is now to be able to grow this. And as I mentioned, there are two main growth processes that we use to expand our tissues. The first of these is what we call interstitial growth. And as this name indicates, this is growth that occurs within the tissue. The green dots in the perichondrium are the mesenchymal cells. So here, we can do that. So, no. Oops, no, hold on. There we go. All right. Interstitial growth, growth that occurs from within the tissue. Remember, one of the things we talked about with our osteoblasts and osteocytes in osteoclasts is that those cells cannot divide. Only the osteogenic cell, only that unipotent stem cell can divide. But the same thing is not true with cartilage. No, well, the groups were assigned last class. So yes, so that was assigned last class. All right, so the chondrocyte is capable of dividing. So here inside of this lacuna, our chondrocytes are going to divide. And at first, they're sharing that lacuna, they're sharing that room. However, as anybody who has two children, two daughters like I do, when they're young, they don't mind sharing their room so much. But what happens when they start to get closer to being teenagers? Do they like sharing rooms anymore?
No.
No. And so these are each going to want their own room. And the way they're going to go about doing that is they are going to start producing more matrix and surrounding themselves with more matrix. And as they produce more matrix and surround themselves with more matrix, what's going to happen is they are going to expand away from each other. By making this new matrix in the middle, they're going to push apart from each other until they're in their own individual lacunas. And so by making this new matrix on the inside of the tissue, we are going to expand the tissue by making new tissue from within. So we're expanding the tissue by making new tissue inside of the tissue. In this way, we can make our chunk of hyaline cartilage bigger. And we'll use this as a growth process to make the original bone model. However, as we've learned, life is lazy. Once it has a good way of doing something, it's not going to want to have to reinvent the wheel. So this growth process is also how we grow bones in length. Laura, you had a question?
Oh, yeah, I was going to ask that. So is it just used for the length or the width?
Well, in this case, these two would be pushing more against each other. So these two would be expanding out more left and right than top and bottom. But
Okay.
in another lacuna, we could have two cells that are expanding more top and bottom and expanding out that way. So yeah, as you have these lacunas, as you have these chondrocytes dividing in the lacunas, they're going to be making more matrix and expanding essentially in all directions. But you're right. In this particular one that I've drawn, it's going to definitely be more left and right than it would be top and bottom. Okay? And that's important because, like I said, that's also what we're going to do when we grow bones in length. When we grow the bones in length, we're going to have them all growing in one direction. So yes, Aravek, to answer your question, yes, in this case, when these chondrocytes divide, they're not just maintaining the matrix, they are actually making more matrix. Absolutely. Making more matrix and expanding this tissue from within.
Do they also separate and have their own lacuna?
Yeah, and they will continue to make matrix until they separate into their own lacunas. Again, if you don't like my pretty picture that goes along with this, your book does have a nice picture that does a good job of showing this. Notice here, you can see how they've got a chunk of hyaline cartilage with one chondrocyte. That chondrocyte divides into two. Those two make more matrix surrounding themselves and separating themselves from each other. And you can see how the tissue expands essentially in all directions. Yes, it's going to go more left and right, but as it's making more matrix, it'll go up and down as well. It's going to expand the tissue from within. Yes, well, I would say that it expands the tissue. It's not so much about, we're talking about width in this case, but it is making more tissue. We had a block of cartilage that was this big before. And now that our chondrocyte has divided and expanded, it's a much bigger block. It has expanded the tissue, right, by growing more cartilage from within.
At what point will the chondrocytes know when to stop expanding? Because kind of like when you're growing as a child, you stop at a certain point.
True, but so what I will tell you, there's two answers to your question. The first is that what drives most of these processes are going to be chemical signals, hormones and other chemical signals that coordinate this activity. This isn't just happening randomly. There are chemical signals and hormones that are driving these processes. However, I will give you a hint at the reason our bones stop getting longer isn't because the chondrocytes stop dividing. So we will actually see why our bones stop growing in length. And it's not because these chondrocytes stop dividing. It's because something else interesting happens. Okay, but we will get to that. We'll build to that. Excellent question, because I love the way you're thinking about it. That shows that you're thinking about it the right way. That is spectacular. All right, any questions? Oops, let's stay here for a second. Any more questions? So again, this is how we grow the tissue by adding new tissue from within the tissue. And this is also how we're going to grow the bones in length. Madison, you had a question?
You just answered my question.
Oh.
I was like, this is the way that we grow the cartilage for long bones specifically.
That is correct. Yep.
Okay, thank you.
I have a question. So with all bone growth, does it have to be cartilage first and then it calcifies into hard bone later?
Yeah. So the short answer to your question is yes. Like everything, it's a little bit more complicated. There are some bones that don't work that way, like some of the bones of the skull or the mandible or things along those lines. But for most bones, yes. And definitely all the long bones. For all the long bones, absolutely. And that's really what we're focused on. We're going to get to the long bones, but yes. So for absolutely most bones, yes. It has to be cartilage first.
I have a question. So
Yeah.
the new matrix that they formed, did those make the new lacunas like for their own? Is that?
Well, notice they're just it's like they're in this. They are they're making new matrix and they're putting the matrix between them. So they're basically like taking building blocks like Legos and using those Legos to build up a wall between the two of them. And as they build up the wall between the two of them, they get pushed further apart from each other until ultimately they're in their own lacunas. Yes.
Okay, thank you.
Yep. Yes, and that's what we're looking at here. We are making that cartilage model for the formation of the long bone. But before we can make the long bone, we have to make the cartilage model. And so notice here, we're expanding the tissue from within, but we have a second growth process we can use to expand the tissue as well. Now for this, I have to cheat a little bit by taking our perichondrium, and I'm just going to cheat and pull it away from the surface. We know it's in direct contact with the end of the cartilage, but I need some room to cheat and play on this so we can talk about the second growth process. And the second growth process is appositional growth. With appositional, oops. With appositional growth, the growth occurs. I want to be consistent. I want to look how I wrote it the last time so I can be consistent. That occurs on the outside of the tissue. Or we can say under the perichondrium. Let's think about what happens here. We have this mesenchymal cell, and this mesenchymal cell can divide, and when it divides, it produces a chondroblast. So let's actually write this out. Mesenchymal cell divides. When it divides, it produces a chondroblast. And what do chondroblasts do?
They form a matrix around themselves on the outside.
Yeah, exactly. They form new matrix. And that's what's going to happen. This cell is going to start to produce some new cartilage matrix until it has completely surrounded itself with matrix. Of course, once surrounded with matrix, what happens to the chondroblast?
Turns into a chondrocyte.
Yep. The chondroblast matures into a chondrocyte in a lacuna. And this just doesn't happen with one. It happens to a second and a third and a fourth and a fifth. And because the perichondrium is still on top and still has mesenchymal cells, a seventh and an eighth, and a ninth and a 10th and 11th and a 12th, and so on and so on and so on and so on. And notice we continue to add more layers under the perichondrium on top of the matrix. So more layers of matrix, so let's say it that way. Under the perichondrium and on top of the tissue. And so in this way, we can elongate. And again, notice I just did it on one side. This can be happening on all sides of this, expanding the tissue. So notice we can expand the tissue two ways, both from within and on the outside. And of course, because life is lazy, this is also how we grow the bones in width. And this is going to be how we form those organizing structures of our compact bone, the osteons. And again, I appreciate my drawing is a little simplistic. So we have the pretty picture from your textbook showing this same thing. Notice here we have our perichondrium, and we have those mesenchymal cells that are dividing, producing new chondroblasts. Those chondroblasts make new matrix and surround themselves with matrix, and we are adding layers of new matrix on the outside of our model, making it bigger. Yes. Yulia, you had a quick question?
Yeah, sorry, I couldn't unmute myself. So is one of them the way babies grow and the other one the way regular bones grow, or are both of them used in both types of growth?
Both of these are occurring in the baby that is growing inside of mom. So again, remember that embryo, when it first forms, doesn't have bones. It has fibrous connective tissues. It has cartilage that we then convert into bone. So what we've done now is basically using these two growth processes, using the expansion of the tissue from within, the adding of new layers on the outside. What we're going to be able to do is make a chunk of cartilage that is the shape of the bone we want, the basic shape of the bone we want. And once we have that cartilage in the basic shape of the bone we want, then we will use endochondral ossification to turn it into a bone.
Okay, so we're still thinking like fetal growth.
Yes, this is all happening in utero. That is correct.
Okay, thank you.
Yep. And again, we haven't gotten to the bone making process yet. This is all the stuff that has to occur before, but it's worth taking the time to talk about because, like I said, we're going to use these growth processes, both of them, a second time in bones, but it gets used in other tissues and other organ systems as well. All right.
I have a question.
Yes.
And so in appositional growth, does the perichondrium, like, do they expand because it gets added on, or is it just gets added onto the layer?
Yes, because remember, so what's happening is, let's actually take a really simple example. So here is our cartilage, and we know it is completely surrounded by a perichondrium. But remember that perichondrium is just a dense irregular connective tissue, right? It's a bunch of fibers at all sorts of different angles. And underneath it is the stem cells. So as the stem cells divide and as those stem cells cause the making of new matrix, oops, wrong color. So the stem cells divide, new matrix is made, and we're adding new matrix on the outer surface. Then our perichondrium is just going to, expand to now surround it as we add new matrix on the inside. And as we make new matrix on the inside, and it causes the tissue to expand outward, right? We're going to have to accommodate it as well. Oops. There we go.
Okay.
So yeah, so that's the nice thing about this dense irregular connective tissue. It will expand and to stay around the tissue as it is being formed. Questions are a good thing. As I've said many times, the only stupid question is the question not asked. So again, the point of you being here is to learn this information. The point of me being here is so that you can ask questions. If I didn't want you to ask questions, then I would have recorded these the week before school started, and I'd still be asleep right now. Right. So the whole fact that we're here doing this live is because I expect it to be interactive and I want you to ask questions. So I love it. So absolutely love that. All right. Excellent. So we have those two growth processes. And like I said, this appositional growth, we're going to increase the width of the cartilage. And like I said, we'll use it to increase the width of bone, and we will use it to make osteons. So again, very, very important process that we will see.
I have a quick question.
Yes.
So is appositional growth, I don't know if maybe I'm a little confused. Does appositional growth form the perichondrium outside, and the, what was it, interstitial growth is what forms the, what was it,
No,
layer
great question.
inside?
No. So in both cases, both appositional growth and the interstitial growth in both of these cases are making more cartilage.
Okay.
So notice here, and again, we'll keep these pictures because I think these pictures are a little bit nicer than mine. Notice here with the dark color, they're showing the new matrix that we're adding on the outside. But at the same time we're adding new matrix on the outside, we're also adding new matrix on the inside. So both of these are processes that we're using to make more cartilage. Because, and I'll give it away for you right now, like I said, the goal of this process, using these two different growth processes, so let me save that, let me clear that, and let me draw this, is going to be to, how do I want to do this? Oh, okay, do it this way. Is to use the cartilage to basically form it and expand it into something that looks like the bone, the long bone we want to make. So this is our starting point. This is hyaline cartilage, right? Which means, of course, that in here are all of the lacunas. And of course, those lacunas have the chondrocytes in them. And of course, on the outer surface, we are going to have that perichondrium. Right, which of course is going to house, I guess I need a different color now for my mesenchymal cells. My mesenchymal cells. So that's the end goal of our two processes. The end goal of these two processes, the interstitial growth and the appositional growth, is to take a small piece of cartilage and grow it into the size and the shape of the bone we need. And once we have the size and the shape of the bone we need, then we're ready for our first growth process. I mean, our first bone making process, and that is endochondral ossification. So now using our two growth methods, we now have our cartilage model. So again, this is going to start with a cartilage model. And we use this to make most bones and all long bones. All right.
That makes more sense. Thank you.
Excellent. That's what I like to hear. I like to hear that things are making sense. Perfect. All right. Questions on this? All right, then let's talk about endochondral ossification. And even though I've just done an amazing job of drawing this, I'm going to cheat and erase it. Do I want to erase it? I've never done it this way. All right, let's try it this way. I am going to cheat and change these a little bit because I don't want to use the red. I don't know if this is going to give me enough room to write around it, but we'll make it work. And if it doesn't work, then oh well. All right, so we'll slap some quick labels on here as soon as I get this all done. There and there. And then we'll make these black. All right, so as we did with mitosis, I want to do the same thing here. I am going to go through this entire process here on the board with my words, with my pictures. And for this part, I strongly encourage you to just put your pens and pencils down, just watch, listen, think about this process. Then we'll come back and do the whole thing again with all the pretty pictures and all the pretty words, and you can write till your heart's content. But if you're writing right now, you're not listening and you're not going to comprehend. So I really strongly want to encourage you to just watch this process the first time. Think about what I'm saying and what we're doing, and then we'll go through it again and you can read it and you can write and do all that stuff as well. All right. So here's going to be one of the keys. We have been making, oh, that's going to be way too big. Cartilage. But now we want to make bone. And I kind of gave you the key to this earlier. If these mesenchymal cells, which are pluripotent stem cells, happily doing exactly what it is that they want to do, which in this case is make cartilage, but we want to make them to make something else, how would we do that? How can we get a cell to do something else? How can we change the environment, change the chemical signal to get it to do something else?
Calcium.
We do know calcium makes cells do wonky things, and that is something that's possible. You got the right idea. It is going to be some type of chemical signal like a calcium or like a hormone. But here's the question. How do we get those things to these cells? How are we going to change the environment?
Well, didn't we have receptor mediated changes within a cell, like when we covered the cell? Would it be the same here?
Well, you've got the right idea. We need chemicals, we need hormones, we need something to trigger some change, but we need something to bring those signals there. And Madison's hit it on the nose, a blood vessel. If we want to change the environment, if you think about it, one of the big differences between cartilage and bone is very vascular and cartilage is avascular. So the very first thing that's going to happen is we are going to have a blood vessel that grows into the perichondrium. That blood vessel that grows into the perichondrium brings new chemical signals, brings hormones with it, brings different nutrients, things like calcium. And what happens here under the perichondrium is now those mesenchymal cells, which were before dividing to make chondroblasts, now divide and make osteoblasts. And so let's cheat. Let's grab my perichondrium and move it away just a little bit here so that when, what color should we use for this? Let's use brown, light brown. This mesenchymal cell's going to now divide to produce a chondroblast. Pardon me, an osteoblast. And what do osteoblasts do?
Make matrix.
What kind of matrix? Bone matrix. There you go. It's not a trick question, right? Osteoblasts make bone matrix. And so it's going to surround itself with bone matrix. When it surrounds itself with bone matrix, it's going to become an osteocyte living in a lacuna. And the process continues. So notice what's going to start to happen is we are going to start to get this layer of bone that is going to start to form on the diaphysis of our long bone. And it's going to occur on both sides. So we're going to actually get the formation of this bone cuff or this bone collar that is going to form on the outer surface. So as a result of these new chemical signals that have been brought in, our mesenchymal cells now make osteoblasts. Our osteoblasts make bone matrix. And we form a bone cuff or a bone collar on the outer surface of the cartilage. Oops. All right. Now, I have one more question. At the beginning of this process, this dense irregular connective tissue right here was sitting on top of cartilage, so we called it a perichondrium. However, now that perichondrium is now sitting on top of a thin layer of bone. So is it still a perichondrium?
No.
Wouldn't it be a periosteum?
Exactly. Our perichondrium becomes a periosteum. Not because it changed chemically in any way. It's the exact same cells, the exact same tissue. The only thing that is different is what it's sitting on top of. But remember, perichondrium is a name based on its location, a dense irregular connective tissue that sits on top of cartilage. Well, now it's not sitting on top of cartilage anymore. Now it's sitting on top of bone. So because it's sitting on top of bone, it becomes a periosteum.
Is that something we should be able to observe, or is that something that we just kind of have to know in our mind?
Well, the fact that it's on, yes, when you see a dense irregular connective tissue on top of cartilage, you know based on its location, it is a perichondrium. When you see that same type of layer on top of bone, you know it's a periosteum. Right? The same way that this soda, because it's sitting on my desk, is mine. If it was sitting on your desk, it would be yours. Right? Nothing about that soda has changed. The only thing that's different is its location. And so we give it a different name based on its location. If it's on your camera, it's yours. If it's in my camera, it's mine. All right. Questions on that? Okay. At the same time that this is going on, there are also changes that take place inside the diaphysis. Those same chemical signals that blood vessel brings into the outside changes what's happening on the inside. Remember before, those cells were dividing and they were making new cells and they were growing apart from each other. But now what's going to happen is our chondrocytes in the diaphysis grow very large and become very active metabolically. So what happens here is the chondrocytes here in the center grow really, really large. Now, of course, there isn't anything inherently wrong with these cells getting really, really large. But obviously, if the cells are large, the lacunas around them have to be large as well. And notice as these cells expand and their lacunas expand, what happens is that our matrix thins. There's now less of it. And it starts to harden. If this matrix thins and hardens, is it going to be harder or easier for these cells to get the oxygen and nutrients they need? Is it going to be harder or easier for them to get rid of their wastes?
Harder.
Harder. So it becomes harder for them to get oxygen and nutrients, harder for them to get rid of their wastes. And so not surprisingly, the chondrocytes die. And when they die, they leave a cavity. And that cavity is the perfect place for new growth to occur. Of course, if new growth is going to occur here, we need a way to bring the new cells, to bring the chemical signals, to bring the other things in necessary to be able to start that new growth. And how might we want to do something like that?
Blood vessel?
Blood vessel, absolutely. A blood vessel grows into the center of the diaphysis. And not just any blood vessel. This blood vessel is what is known as a nutrient artery. The nutrient artery grows into the diaphysis. When it grows into the diaphysis, it brings new chemical signals, it brings new cells, and in particular, it brings osteoblasts. And what do osteoblasts do?
They make bone matrix.
Make bone matrix. Now, we know bone matrix can be organized in one of two ways. It can be perfect concentric circles around a central canal where they're making these beautiful osteons, or they can be all regular at all sorts of different angles and orientations. What do you think is going to happen here in the center? Are we going to get perfectly precisely arranged bone matrix in here?
No.
No. So what happens is we get a small chunk of spongy bone that forms. And this small chunk of spongy bone that forms is called the primary ossification center, because this is the first place we're making bone in the cartilage model. So we get this chunk of spongy bone that forms here in the center. Well, but remember, we've just made a hollow cavity and blood vessels are persistent. So, and again, it's not bone, it is, there is a thin layer of bone, but it's a thin layer of bone. If we were in the classroom, one of the things that I would encourage you to do, if you ever get the opportunity to look at a real bone, and I'll be honest, I've never actually looked at a chicken leg bone to see if it's there, but it might be. But when you look at the human bones, and you may see it on some of the pictures, like in the atlases and things like that, you'll notice in the center of diaphysis is a small hole. That small hole is actually the hole where that nutrient artery grows into the center of our diaphysis. And so all long bones have that. Again, these are I wouldn't say programmed, but they are directed by chemical signals. These blood vessels grow where they're supposed to based on the chemical signals that are given told them to do that.
And this is where those perforating canals come from, yes?
Eventually, the perforating canals are actually going to be coming more off of the periosteal blood vessel.
Oh, okay.
That is going to form. Yep.
So is the growth of the chondrocytes first, it forms the spongy bone, and from there it will form like the medullary cavity?
We're going there next. You've absolutely got the right idea. But also remember, it's the osteoblasts that are making this primary ossification center, not the chondroblasts. But yep, you got the right idea. We're going to get there because you're right. You've hit on a problem. This expansion continues. Right? Our spongy bone is going to grow towards both ends. And we start to, oops, this shouldn't all be capitalized. And we add layers of organized compact bone on the outer surface. So what's going to start happening here is that and I'll cheat, use a bigger line. Out here on the outer surface, we're going to start to get some bigger, thicker, compact bone on the outer surface of our diaphysis. And on the inside, our spongy bone is going to continue to expand and grow throughout the entire diaphysis. However, you've hit the nail on the head. We know that the inside of the bone of the long bone shouldn't be solid spongy bone. So we need to hollow out the medullary cavity and add the red bone marrow. If only there were cells that we had that were capable of breaking down bone matrix, we may be able to hollow that out. Do we have a bone like that?
Yeah, the
Or a cell
osteoclasts.
like that? There you go. So osteoclasts form, and those osteoclasts that are going to form, they break down most, but not all, of the spongy bone. So now we've got a thick outer layer of compact bone, a thin layer of spongy bone on the inside, and a medullary cavity in the center that will contain red bone marrow. And now our diaphysis looks exactly like a mature bone. At this point, we are now done developing the shaft of our long bone. Again, remember, this process starts early. It starts around week 6 or 7. When do you get that first really good ultrasound? And I'm not talking about the one where it looks like a little peanut. When do you get that really good one where you see the fingers, you see the hands, you see stuff like that? About 20 weeks, right? By 20 weeks, you can see the ribs, you can start to see the arms, the legs, the fingers, stuff like that. Well, we're going to have a little bit. Remember, we do have little bone marrow in there. Remember, the yellow bone marrow doesn't come to a much more mature. After adolescence, we slowly start to develop a yellow bone marrow. In an adolescent, in a baby, and certainly and even in an adolescent, all of the bone marrow is red bone marrow. We don't start producing yellow bone marrow till later on. And it isn't so much that we don't want it to have spongy bone in the middle. It's just it doesn't have spongy bone in the middle. It's filled with that bone marrow. And we need that bone marrow to make a lot of blood cells. So that's probably more why. You know, why is one of those tricky questions, but that's there. But the point is, at this point, like week 20, our diaphysis of our long bone pretty much looks like a mature diaphysis is going to look. All right. Madison, you had a question?
So because we only have red bone marrow and like from this point and on throughout adolescence and we don't get yellow bone marrow, is that why when it comes to like a kid breaking their leg or something, it's not as severe when it comes to recovery time? Because the red bone marrow can supply a faster...
Oh, I see where you're going with that. I would say it probably has more to do with, remember we talked about the overall metabolism. We'll talk about it more as we move forward. The overall metabolism of the cells. Our bone cells are much more active at building bone when we're younger than they are when we're older. So the fact that our metabolism is set up in such a way where they're much more active in building bone matrix, that's why it heals more rapidly.
Yeah, because like I work in a PT office, and so dealing with our older patients, they're there much longer than our younger ones when it comes to recovering from this. Like we could have two of the same surgeries, but one patient being younger, and the younger one typically leaves soon.
Yeah, typically because they have a higher metabolism for these cells, they repair more.
Okay,
absolutely.
So that confirmed my beliefs.
Bones definitely do adapt to the stress that we put on them. And like I said, we'll talk about that more. I promise we'll get to that. We'll get to that next week, but we will definitely talk about that. But the short answer to your question is yes, bones adapt to stress. So the more stress you put on the bones, the stronger those bones get. So someone who is slightly heavier can have stronger bones because of that, but they're also putting more stress on their bone, which could also cause damage as well. So I wouldn't say that necessarily a slender person has weaker bones. I think they both have bones appropriate for the stress that you're putting on them. All right. So this starts early. And like I said, by week 20, we start to see the long bones looking like we expect them to see. But notice we're not done. We haven't done anything to the epiphyses yet. And here's the good news. The development of bone in the epiphysis is similar to in the diaphysis. However, right, so let's start with how they're similar. Obviously, we need to have the chondrocytes enlarge. And die. Once they enlarge and die, there is then a space available and a blood vessel grows in. This blood vessel is actually going to grow in through the metaphysis. Oops, wrong color. This one grows in through the metaphysis. So it's known as the metaphyseal blood vessel. Grows in, bringing new chemical signals, and we bring in osteoblasts, and we form a small chunk of spongy bone. And guess what we call this small chunk of spongy bone that is going to form in the diaphysis? I mean, in the epiphysis.
Epiphyseal, probably something.
Well, wasn't the first one the primary ossification center? So what do you think the one that forms second is called?
The secondary.
Secondary.
Yeah, see how easy that is? So the process is going to be similar. Chondrocytes enlarge and die, making a cavity. Blood vessel grows into that cavity. Osteoblasts form, and we start making spongy bone. But while these processes are similar, there are also some big differences. For starters, how many secondary ossification centers do we make?
2.
2, right? 1 primary, 2 secondary, which makes it real easy to remember, right? Because you, after all, have 1 diaphysis, but you have 2 epiphyses. So you are going to form 2 secondary ossification centers, where you only form 1 primary ossification center. So one of the big differences is, oops, do this again, form 2 secondary ossification centers. Second big one, which I should have probably said first, is the timing of this. These secondary ossification centers typically do not form until just before birth or shortly after. I am not suggesting you grab a baby and twist them in a pretzel, but has anyone ever felt and played a little bit with a baby's hand when it first forms? Again, I'm not encouraging you to do this, but you can pretty much take their fingers and bend them all the way back to their wrist, right? They are pretty bendy. And the reason they're so bendy is their epiphyses haven't formed yet. So the timing of this is much, much later. We're going to form 2 of them. They occur much, much later than the first ones did. Now, like the primary ossification center, this is going to expand within this space. However, as it expands into this space, do I want to then bring in osteoblasts and hollow it out?
No.
No. So in this case, it stays spongy bone. no cavity forms. So that's a big difference. And here's another huge difference. Not all of the cartilage is converted into bone. Some of the hyaline cartilage is going to stay behind on the outer surface of the epiphysis. Why would we leave some hyaline cartilage on the outer surface of the epiphysis?
Because that bone is most likely going to articulate with another bone.
Excellent. And so the hyaline cartilage at the end of the bone where it forms the joint, we would call what?
Articular cartilage.
There you go. So some is going to stay on the outer surface to form that articular cartilage to form the joint. Excellent, excellent. And the second place we're going to leave some hyaline cartilage is in the metaphysis. Why would we want to leave some of this in the metaphysis?
That growth plate or the plate.
It forms that growth plate for the bone growth in length, but we want to use appropriate anatomical terms. So we're not going to call it the growth plate. What are we going to call it?
I think it was a epiphyseal plate.
Yeah, that is absolutely correct. So it is going to form the epiphyseal plate. And let's put it on both sides. Our epiphyseal plate, our articular cartilage, and now we have a fully formed bone. Our epiphysis looks like the epiphysis is supposed to look. Our diaphysis looks like the diaphysis is supposed to look.
Are you going to post this picture?
Yeah, I can post this picture. And like I said, I've also got the pretty pictures from your textbook, which do a really nice job of showing this as well. All right, questions on this?
No.
All right, excellent. So here's what we're going to do. I promised you we're going to go through it twice, but because of the timing and the break and everything, yes, so the development of the secondary ossification centers, the forming of the bone in the epiphyses, in the heads, occurs either right before the baby is born or right after the baby is born, depending on which bones they are. Some long bones form their heads a little bit earlier, some a little bit later, but they're all right around the time of birth. So very, very different timing for when the diaphysis forms versus when the epiphyses form. All right, excellent. Let's go ahead and take our first break. And then we come back from that break, we will then go through this whole process again with all the pretty pictures and all the pretty words from your textbook, make sure we understand it. And then once we have that, then we'll talk about our second process. And that second process will be intramembranous ossification. And then from there, I think that's all I want to cover for today. Then we'll do our introduction to the bones and then start our group presentations. All right, excellent. So let's go ahead. It is 1:20. We'll take a 15-minute break. So that means coming back at 1:35. And so we will restart at 1:35. Oops. And I will start the recording at that point. All right, so any questions before we take our first break? Oops, I already used that. So let's use that. All right, see you guys in 15 minutes. You guys have had a little bit of time to sit and think about this process. Let's go ahead and go through it step by step here. So again, we start with our hyaline cartilage model that has that perichondrium on the outer surface. And we need to change the chemical signals. We need to change the environment. We need to change those mesenchymal cells from growing chondroblasts into growing osteoblasts. And the way we do that is by changing the chemical signals, by growing a blood vessel into the environment. And the first environment we grow it into is the perichondrium. When we grow it into the perichondrium, now these new chemical signals that are being brought into the location cause the mesenchymal cells to form osteoblasts. And osteoblasts do what osteoblasts are going to do. They make bone matrix. And so we get the formation of this thin shell, this bone cuff or bone collar on the outer surface of the cartilage model. And because we love our vocabulary, as we talked about, that perichondrium that was sitting on top of cartilage is now sitting on top of bone. And because it's now sitting on top of bone, what does our perichondrium become?
Periosteum.
Periosteum. Excellent. Perfect. All right. Questions on that? All right, excellent. At the same time that we're seeing those changes that are occurring on the outside of the diaphysis, there are changes that are going on in the inside of the diaphysis. Those chondrocytes, which before were dividing and expanding the tissue, now instead get really, really large and really metabolically active. And so as these large chondrocytes start to form in the middle, that causes the matrix to thin, and it causes the matrix to basically harden. Now those really active chondrocytes can't get the oxygen and nutrients they need. They can't get rid of their metabolic wastes. And as a result, they die. And what they leave behind is just this hardened cavity, which is the perfect location for growth to take place. Of course, if we want to start making bone in that cavity, we need to bring new cells, we need to bring new chemical signals, we need to change the environment. And how are we going to change that environment?
Blood vessels?
Yeah, blood vessel growing in, absolutely. And that's exactly what happens. That nutrient artery invades that space, burrows itself into that cavity, bringing new chemical signals, bringing new cells, and forming osteoblasts. And those osteoblasts form a small chunk of spongy bone that we call our primary ossification center, because it is the first place inside the cartilage where we're making bone. That spongy bone expands towards the two ends, filling the diaphysis with spongy bone, which is great because we got bone, but that's not what we want our mature long bone to look like. So we basically need to remodel it. That remodeling basically involves two main processes. While we convert all that cartilage to bone with osteoblasts, we are then going to bring in osteoclasts that destroy, and I want to make sure we emphasize this, most of the spongy bone. Remember, there is still going to be a little bit of spongy bone lining the inner cavity. This hollows out the inner cavity that we call the medullary cavity, and we're going to fill it with red bone marrow. At the same time, we see that the outer surface is forming this thick, compact bone on its outer surface. And now our diaphysis has the mature shape that we want. And like I said, for many of these bones, this is finished by week 20 or close to it. So this occurs really, really early. The reason that's significant is because notice nothing has happened in our epiphyses yet. Our epiphyses are completely unchanged and will stay unchanged for weeks. It's not until right about the time of birth, or even in some cases shortly after it, that we start to remodel our epiphyses. Hold on. So most of it, so the point I was trying to make, if we go back, although this picture shows it as well, notice there is still a very thin layer of spongy bone that lines the inside of the medullary cavity. So as you look in a cross section through a long bone, you see that there is a very thin layer of compact bone lining the inner surface. So not all of the spongy bone is removed, just most of it. So yeah, that was the point I was trying to make. And of course, most of that spongy bone is removed by the osteoclasts. All right, so at or near the time of birth, the exact same process starts to occur. Our chondrocytes enlarge and die. A blood vessel grows in, and I hate that our artist has put it way up here on the epiphysis. It is much more appropriate that it is going to grow in down here from the bottom, more closer to the metaphysis to grow in. But again, artistic license. Starting a chunk of spongy bone. These two chunks of spongy bone are called the secondary ossification center, and not just because they are two of them, but because they come second. So the process starts the exact same way. Chondrocytes enlarge and die, cavity forms, blood vessel grows in, we make spongy bone. They start the same way, but there are some big differences in the two processes. Right here, the spongy bone that is made is going to stay spongy bone. We don't hollow it out. We don't make a cavity in the epiphyses. And we don't get rid of all of the cartilage. That hyaline cartilage is going to remain in two main locations. On the outer surface, forming the articular cartilage, which is, as you guys pointed out, where we're going to form the joints, and the epiphyseal plates, or as someone commonly referred to them as the growth plates, but don't call them growth plates on the exam, the epiphyseal plates, which is where our bone is going to grow in length. And so with that, we have made a long bone. And like we also talked about, this is going to be the process that we're going to use for most bone formations. Most of the bones in your body are made in this fashion. But as we know, most is not all. So we do have a second ossification process we need to talk about. All right. Questions on this? All right, excellent. Apparently everybody who was asking questions fell asleep, but all right, excellent. Well, again, the nice thing about this, we are going to cover a lot of material today, but I will remind you that we don't meet again for 5 days. So you have plenty of time to go back and synthesize this information and make sense of this information. So it's okay to be a little bit overwhelmed, but this is the pace of the class. So, but that's the nice thing about with the recordings, as someone mentioned, you get the chance to go back and revisit them as many times as you want. You have 5 days to go back and study this. And so absolutely, definitely spend more time with this material. All right, excellent. Now, this kind of gets to the question someone was mentioning before. Notice we talked about how, and again, a lot of it, one of the things I love about anatomy and physiology is I don't think it's necessarily inherently tough. It's just a lot of information. But if you also break it down, you can see that this whole process of making the bone is really the same thing that happened 3 times. A blood vessel grew into an area, changed the environment, changed the chemical signals, changed the cells, and we made bone. First, it happened out here in the periosteum. Then it happened in the diaphysis, and then it happened in the epiphysis. So basically, it was the exact same process that happened 3 times. A blood vessel grew in, changed the chemical signals, brought new cells, made bone. But notice the blood vessels from these 3 environments all grow together. So here, someone was talking about the blood vessels of the periosteum. You can see how they're going to form the perforating and the central, the blood vessels in the perforating canals and the central canals. You can see how the nutrient artery is going to communicate with the metaphyseal arteries. And like I said, when you get the chance to look at a real bone, you will actually see these holes where the blood vessels pass through. And they all grow together into this huge, highly vascularized structure, right, with all 3 of those. And even though this picture just shows the arteries and the veins, what else do we know are going to travel right along, right next door to those arteries and veins?
Nerves.
Nerves, absolutely. So nerves and also lymphatic vessels are going to follow these exact same pathways as well.
What's the reason for nerves having to be there?
So the couple of reasons. One is that it the bone cells can be influenced by electrical activity, and so that can influence that. And then the other reason is so that when it gets damaged, you're aware, right? When you break a bone, it hurts like heck. And you want that information so that you know not to be able to use it and continue to damage yourself.
True.
All right. So that is endochondral ossification and our end result. But as we talked about, there is a second method for making bone. So let's again, this one's not quite as elaborate, but I still think it's worthwhile to take the time to draw it and to talk about it. So let's do that and make sure I've saved this so that I can go ahead and clear that. This second growth process is intramembranous. Ossification. This is how, for instance, we form some of the flat bones of the skull, mandible, things along those lines. And as the name indicates, here we are making a bone inside of a fibrous connective tissue. Now, notice I didn't say dense regular or dense irregular or elastic or something like that, because this is an embryonic tissue, and many of the embryonic tissues are different from the tissues that we have as adults, but it's still the same general idea. We have collagen fibers that are going to be located. Actually, I don't want to use black for that. Let's use a lighter color. There we go. So here's some collagen fibers that are going to be all over the spot. That's way too uniform, but that's all right. And inside here, of course, we are going to have mesenchymal cells, and we'll make those green as well. So this is our starting point. We have this fibrous connective tissue with all these collagen fibers inside of it and all of these mesenchymal pluripotent stem cells. So again, this is a collagen fiber. And these are mesenchymal cells. All right. So what happens first here is that the mesenchymal cells will clump together and start to divide to form osteoblasts. So right here in this location, and I'm going to cheat and just draw it with a circle. So in this location right here, this mesenchymal cell is going to start to divide to produce a couple of osteoblasts. And of course, those osteoblasts are going to surround themselves with matrix. And as they surround themselves with matrix, they're going to mature into our osteocytes inside of a lacuna. Now, we've seen this before. This is a small cluster of cells forming a small clump of bone. And so basically, we form an ossification center. However, do you think just one of these is going to form just in this one location all by itself? No. So what happens is many ossification centers form at the same time. And again, it's at the approximate same time. It's not like they all instantly appear at the same time, but the key is that many of them form and they don't have a sequence the same way that we see in the long bone. So again, for simplicity, another ossification center is going to grow here, and one's 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 going to grow here. So many of these small little ossification centers are all going to form within this fibrous connective tissue. So we don't have to worry about a primary, secondary, tertiary, quaternary, and so on and so forth. There are just many of these forming all at the same time, approximate same time, within this fibrous connective tissue. Now, as these small ossification centers form, they're, of course, just like we saw in the cartilage, going to expand and grow beyond. And as they grow out from the center, they're going to connect to each other. Yeah, the brown circles represent the ossification centers. No, those are the ossification centers. The little dots were the cells. I just didn't want to have to draw a ton of little dots to do that. Yep. So this one, and now I'm going to cheat and just use my highlighter again. This one's going to grow out and expand this way. Yeah, Ervik, you had a question?
Yeah, when mesenchymal cells divide, aren't they going to make osteogenic cells first, then?
Yes, you are correct. They are going to make osteogenic cells, but then the osteogenic cells make the osteoblasts. So you are correct. I have not been mentioning that part of the process because osteogenic cells can only become one thing, osteoblasts. And it's the osteoblasts that are making the bone that we care about and that we worry. But you're absolutely correct on that. Yep, thank you. All right, perfect. So these are going to continue to make bone layers of matrix that are going to expand out. And as they expand out to each other, they're going to start to connect to the ossification centers that are surrounding them. And notice, as these are expanding out and growing, two things are happening. Well, let's actually do it first. As the ossification centers grow more bone and expand, they connect to nearby ossification centers. And as they connect to other nearby ones, notice they're making a bunch of irregular points and processes. And of course, those irregular points and processes are what we call the trabeculae of the spongy bone. So we get to start getting these irregular points and processes, these trabeculae that are forming as a result of the expansion of these ossification centers. Okay. Now, as the trabecula form and the bone tissue expands, two things occur. If you think about it, as this tissue is expanding outward, it's going to be pushing on the fibers that are around it. So those fibers that are around it are going to get pushed up on, and those fibers are going to start to condense around the outer surface of this bone as it is expanding. And as it extends out, and let's actually change the color a little bit so it's a little easier to see. As it expands out on the outer surface and condenses down, it's also going to be capturing those mesenchymal cells here along the outer surface of this bone that is forming. So now I have a bunch of irregularly oriented collagen fibers on the outer surface of my bone matrix. What would I call something like that?
Is that the periosteum?
It's exactly what it is. So notice as the tissue expands, our periosteum forms. The periosteum forms on the outer surface. At the same time that that periosteum is forming on the outer surface, we know there are all these spaces, all these cavities between the trabeculae. And as we just learned, we know that blood vessels just love to grow into cavities. So blood vessels are going to grow into the spaces within the trabeculae, and start depositing red bone marrow. So we're going to have some blood vessels that are going to grow into these spaces. And as they grow into these spaces, they're going to start to fill these spaces with red bone marrow. And lastly, the periosteum ends up organizing the cells, the mesenchymal cells, on the outer surface of the bone. And because they're more organized now, we can start making a thin layer of compact bone, which of course, remember, because we're dealing with flat bones and skull bones here, we would call the cortex, starts to form. So on this outer surface, we are going to get the formation of a thin layer of compact bone. And while this is mostly good, is this necessarily going to have the precise shape that we want for this bone? Is this the exact shape of the bone that I'm necessarily going to have in the skull by just the random formation of these ossification centers?
No.
No. So then the last thing we need to do, we need to remodel the bone into shape. And we will do that using osteoblasts and osteoclasts. So maybe we don't necessarily need it as elongated as it is there. So we can start to use some of, and here, let's use a white. I can start to shade down, use my osteoblast to cut this down a little bit and reshape it into the form that we want. Or maybe I need to add a little bit more enlargement over here so I can use my osteoblast to build this side up. So I can build up on some sides and I can break down in other sides so that I can get the precise shape I want using those osteoblasts and those osteoclasts. Yes, Laura.
So when say like when a baby has to have a helmet on for a couple time or for a short amount of time, is it because the osteoblasts and osteoclasts didn't really form their skull right, or is it completely different?
No, it's typically something different. So, and then again,
Okay.
It also depends on your timing and when you're talking about these things. You have to remember that all babies are born with the skull bones of their head not fully fused together. That's what allows the baby's head to modify its shape to be expulsed through the vaginal canal. Right. So that ability to have that little bit of flexibility and give. And what's the name for those really soft spots where the bones haven't fully fused together? Anybody know? I know there's somebody out there who knows the name for them. They're called the soft spots, but if you've ever looked at them, you can actually see the blood, the blood pressure change and the little bit of a pumping of them. They almost have a fountain-like appearance to them because they're called the
Fontanelles.
Fontanelles. There you go. Exactly. So that is the case. So every baby is born without the skull bones fully fused together to allow the head to be more flexible to be passed through the vaginal canal. In some individuals, because of developmental delays and things along those lines, there can be a delay in the growing together of the skull bones, or the skull bones may not be as strong or maybe a little softer. And yes, in those cases, those kids do sometimes have to wear protective gear for a couple of years or a year or so while it gains strength that way. So yes, so it's something that everybody deals with their babies when they're first born. But yes, there are some that can have developmental issues that can cause it to be prolonged. Laura, did I answer your question, or do you have a new one?
Oh no, I forgot to.
Okay, no problem. I usually try to put them down, but sometimes I forget, so I wasn't sure if that was a new or an old one. All right. Again, this picture is even uglier than the last one that I drew, but luckily we have the pretty pictures from your textbook that show this as well. So again, here we see a clustering together of those mesenchymal cells and dividing within the collagen fibers to form a chunk of immature osteoblasts. Again, we are making these inside of a fibrous tissue. We're not using cartilage. Like I said, the flat bones of the skull and the mandible are bones that are made this way. And it starts with not one, not two, not three, but many ossification centers all forming together. Our mesenchymal cells cluster and they form osteoblasts. An osteoblast is going to do what an osteoblast is going to do. It is going to make matrix, surround itself with matrix, and mature into an osteocyte. Yes, I can go back to my my Pollock-esque drawing here of this process. Yep. So not one, not two, but many ossification centers form as we start producing these layers of matrix. Right, little bit of spongy bone. Well, again, I'm not the best artist, but I think sometimes the nice thing about hand drawing these things is you can focus on just drawing the parts that are important, and we don't have to worry about all of the artistic representations that go along with it. The osteoid is just the little pieces, that's the little chunk of, remember we talked about how ossification is where the osteoblast is taking the calcium, taking the phosphate, taking the carbonates and depositing it on the collagen fiber. Those are those droplets that are formed are the osteoids that then fuse together to form the matrix. It's, again, they're here, but it's not a vocabulary term you're responsible for, so you don't have to worry about it. Those small ossification centers start to fuse together. And as they, well, yeah, it's the deposited crystals that are being made by the osteoblasts. They're from the osteoblasts, right? Before they surround themselves with matrix. They're little bits of matrix being formed by the osteoblasts. But again, it's never going to be an answer on one of the exams, lab or lecture. Osteoid is not a term you're responsible for. Those small ossification centers fuse together, forming the irregular points and processes of the trabeculae, making our spongy bone. And notice once we start having this spongy bone, we can see two things occurring. Notice they don't show us the collagen fibers anymore, but we can see how all the mesenchymal cells are being shoved up against the surface of the bone as we're forming that periosteum. And we can see the blood vessels growing into the spaces of the trabeculae, depositing our red bone marrow. This formation of the periosteum allows for the organized formation of a thin layer of compact bone on the outer surface, that cortex. And notice, because we were talking about how this is the flat bones of the skull, we get to use our fancy term for the trabeculae, which are the diploe. Then again, using our osteoblasts and osteoclasts, we can then form this into the shape that we want. All right. And there you go. Our second process for making a bone, intramembranous ossification. Questions on that?
No.
Excellent.
So just real quick, that term diploe is only for the skull?
No, diploe is the locational term they give trabeculae inside of flat bones. So any flat bone, the trabeculae inside are referred to as diploe. And like I said, I have no idea why.
But not in the flat bones, what are they referred to? Just as trabeculae?
No, so in a short bone, in an irregular bone, in a sesamoid bone, in a long bone, like the heads of the long bone are filled with spongy bone, but we don't call they're just trabeculae. In a short bone, they're just trabeculae. In an irregular bone, they're just trabeculae. In the sesamoid bone, they're just trabeculae. But for whatever reason, and I don't have a good reason for why, but it is a term we need to know, a trabeculae in a flat bone is also referred to as a diploe. So why it's not a diploe in everything? Because there's no difference in the trabeculae in a short bone or in the head of a long bone than there is in a flat bone, but for whatever reason it is. Well, this particular one, we know it's a flat bone because they called it a diploe. And we know this intramembranous ossification only occurs in flat bones. So if this process is taking place, it's forming diploes because it's doing it in flat bones. All righty. Now, because you guys are out of questions, that of course tells me that you have of course mastered this information, which tells me that we can go on because again, being the smart, sophisticated students that you are, anytime we have two related processes, how many possible essay questions could we have?
2.
3.
3, absolutely. The first one would be describe endochondral ossification. The second would be describe intramembranous ossification. And what would possible essay question 3 be?
Difference or similarities?
Yeah, the comparison between the two. Absolutely. Now, I saw this on some of your exams, so this is the other reason. Aside from this helps us to make sure we understand these, but I think it also helps us to be successful on the exams, is when I ask you to compare two processes, right? I want it to be a direct comparison of apples to apples. I'm not asking you to describe process A and process B, right? That's not what I'm asking for. Right? Or if I'm asking you to describe Laura and I, right? One of us has a beard and the other one's left-handed, right? That's not a direct comparison, right? Saying one's left-handed, one's right-handed, or one has a beard and the other doesn't, those are comparisons, right? But if I have a beard and she has pierced ears, that's not the way that, that's not a comparison. You are not required. Endochondral ossification is the other one. You are not required to set up your essay questions this way. And again, it doesn't have to be an essay question. You can do this, again, at bullet points or whatever it is, paragraphs. But if you have trouble with these types of questions, one of the ways that you can set this up for yourself to help you with this material is not just to have a column for intramembranous ossification and a column for intramembranous ossification, but to also have the characteristic you are using to compare them, to make sure that you're comparing to apples to oranges. So somebody give me a comparison, give me a characteristic, give me a way intramembranous ossification and endochondral ossification are different.
Type of bone.
Okay, excellent. Perfect. One characteristic is the type of bone they form. Intramembranous ossification of forms of what kind of bone?
Flat bones.
Flat bone.
Flat bones, primarily of the skull. Whereas endochondral ossification produces, of course, most bones, but what we care most about, absolutely, as Ariana pointed out, are the long bones. Excellent. Perfect. Give me another way they're different.
How they start out. Like
Okay.
The first one, endochondral, is from hyaline cartilage. The other one is not.
Starting tissue that we are going to convert into bone. Absolutely. And again, saying one is something the other isn't as good as saying what? What is the starting tissue in intramembranous ossification? And again, anyone can answer this.
Fibrous
fiber.
tissue.
Yeah, fibrous membrane. Whereas what is the starting tissue for our endochondral?
Cartilage?
Is it cartilage?
And you get partial credit for that.
Island cartilage.
There you go. Excellent. Give me another one.
The way trabeculae would be called, diplo.
True. Okay, so that is a good anatomical difference, but I'm not sure it's a difference in the of the formation process. It is that's more when we're identifying long bones versus flat bones. That's one of the ways we do them anatomically, but that doesn't really compare the way they form.
I would say the difference of ossification centers.
Okay, excellent. And how about like my number, right? Is that
The
what you mean?
number, yeah.
How many total ossification centers do we have in endochondral ossification?
3.
3 total, right? And also, if you think of the timing of them at different times, right? Dramatically different times when they form. So not only are there 3 total different, but they form at different times. Whereas here, how many do we have in intramembranous?
Multiple, all at the same time.
Many, absolutely. And all around the same time. Now, Ariana, I like where you're going with that. I would say that both, because we're making bone, we have to have collagen fibers. But I think what you mean by collagen fibers, or what I'm hoping or that you mean, is the formation of the periosteum, right? Because in endochondral ossification, what comes first? The dense irregular connective tissue housing immature cells or bone? Which comes first in endochondral ossification?
Let's say bone.
Does the bone come first?
Wait, no, sorry.
Dense irregular?
Dense.
Yeah, I mean, technically it's a perichondrium, right?
Yeah.
At the time, it's a perichondrium, but that periosteum in the guise of the perichondrium is there first, right? We have a dense irregular connective tissue housing immature cells that is there before the bone. Whereas in intramembranous ossification, the bone forms first, then the periosteum forms around it. Is that kind of what you meant, Ariana?
Yes, that's what I meant, yeah.
Excellent, perfect. Along those same lines, how about blood vessels? Do blood vessels grow in before the bone or after the bone in intramembranous ossification?
After the bone?
After.
After bone forms. What is that the same in endochondral ossification?
No.
No, before.
Happens
Comes
during
before.
or
Right. And I'm sure there's easily a dozen more of these that we could do. But hopefully this gives you the idea of what it is that I'm looking at with these type of comparison processes. Notice I'm not describing the processes step-by-step, word for word. I'm directly comparing characteristics from one to the other. All right. Similarities are easier. It's much easier to tell things that are similar. But I think where people get into trouble is when they're contrasting them, when they are trying to identify the differences between them. And so, like I said, you are not required to do this. You don't have to have anything over here you do not have to have this over here on the left-hand side. If you just wanted to give me a list like this, 1, 2, 3, 4, 5, 6, whatever it was that I gave you, that would be perfectly acceptable. All right. I would accept that as an answer. However, for me, organizing it by characteristic helps me to make sure that I'm truly comparing apples to apples and oranges to oranges. So it's an organization thing only that helps me. If it helps you, great. If not, don't do it. Just make sure you provide me with the information. And the only other thing I would say is don't memorize this word for word and put it out this way. Make sure you're expanding upon this as well. Right. Obviously, I'm abbreviating a bit, but if you haven't figured it out yet, one of the things that I'm really, really looking for on the exams is specificity and detail. All right. I know for some of you, that's a very different style. Some of you have had instructors before that want you to read the whole textbook cover to cover. And so they give you this huge, massive breadth of information, but because you're reading the whole textbook, they just want you to have a really superficial understanding of it. I'm the exact opposite. I'm going to tell you precisely, not everything in the textbook, but the small amount of the textbook that I think is most important. But the stuff that I think is most important, you need to know everything about. So I'm looking for depth. I am looking for deep understanding. And the way you show that is with specifics, being descriptive, giving me details. All right. Questions on that? And again, if that's your learning style, you're welcome to do that. But just know that not everything in the textbook is going to be equally emphasized on the exam. The things I talk about, the things you do for homework assignments are the things that, and that is the problem. The problem with trying to read the whole thing is if it's too much information, then you're filling your brain with information that you don't need. So again, in some cases, you have to modify your learning style to match what it is that the instructor wants. I don't see the textbook as a romance novel that needs to be read cover to cover. It is a tool. It is an instrument. If you don't understand endochondral ossification after I've described this to you, then go read it in the textbook, because sometimes seeing the different pictures, hearing it in a different voice can help you to make sense of something that doesn't normally make sense. Some people like to know it all. They need to see how all the pieces fit together to make sense of things. Other people's get overwhelmed by too much information. So again, there isn't just one learning style to be successful, but everybody has their own learning style that will allow them to be successful. And this is the time, if you haven't already, to figure out yours. All right, how are we on time? Excellent. All right, perfect. Questions on this? All right, that is everything that I wanted to do for the lecture. Before we go any further, because we're still doing good on time, we are going to break up and do our group presentations next. We'll take our break first, and I'll break you guys into your groups. So for those people who are presenting today, and again, those people are the people who are doing the skull, we'll have a chance to talk and prepare and everything to do that. So we'll take a break. We'll take some time to do that. But before that, I want to do a quick introduction to what we're talking about, because you guys are going to be focusing on the bone features. So I want to talk a little bit first about the bones. Of course, we are talking about the skeleton, and the skeleton basically is divided up into two main components: the axial skeleton and the appendicular skeleton. What are the structures of the axial skeleton?
Body.
Well, be more specific.
The skull and like the vertebral column.
There you go.
Your rib cage.
There you go. And basically the rib cage. I like it. Excellent. These are indeed the structures of the axial skeleton. Here we see the picture where they're highlighted in color. And of the, how many named bones do we have in the body?
206.
206. 80 of them are in our axial skeleton. So again, we are going to be responsible for identifying these 80 bones and your list of bone features associated with them. And like I said, today we are going to start with the skull. So let's start easy. The skull is comprised of a total of 22 bones. Oops. Those 22 bones of the skull are further divided into the bones that form the cranial cavity. And what is the function of the cranial cavity again?
Protect the brain.
Protect the brain.
Protects the brain. Excellent. And there are 8 bones that form the cranial cavity. And then we also have the facial bones. And because I can do math, that tells me there must be 14 facial bones. And of course, what do the 14 facial bones do?
What makes the
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 face
shape of the
face?
face.
Yeah, they make the face. See how easy that is? Excellent. So identify for me the 8 bones that form the cranial cavity.
The frontal,
All right, frontal.
occipital,
Say again.
occipital,
Okay.
peripheral.
It's not
No.
peripheral. What is
No,
it? Anyone?
it's not
Parietal.
parietal. Thank you.
What else?
Orbital.
Temporal.
Temporal.
Zygomatic.
Nope, not the zygomatic.
Sphenoid.
Ethmoid.
Enoid and the ethmoid. Excellent. Oops. Excellent. But wait, that's only 6. So I must be missing 2.
The
Nasal cavity?
no, 2 of
The
them are paired.
parietal.
What about the parietal?
It's a
The
pair.
parietal and the temporal have 2.
There you
One on
go.
each.
These are paired bones. So there are 2 of them. Excellent. All right, so these are the 8 bones that form our cranial cavity. Notice something about them. The paired bones, the parietal and the temporal, are lateral, away from the midline, whereas all of the single bones are either on or across the midline. All right, perfect. Let's do the same thing with the facial bones then. I heard someone say zygomatic already, so let's cheat and start with that one. And now we know the trick. Is the zygomatic going to be paired or unpaired?
Paired.
Paired, so times 2. I see someone wrote maxilla. And again, with the maxillary bone, single or paired?
Paired.
Paired,
Paired.
excellent. What else?
Man.
Mandibular?
The vomer.
Paired or unpaired? Hold on, don't get ahead of ourselves. Mandible, paired or unpaired?
Unpaired.
Unpaired, excellent. And then you said, what did you say?
I said the vomer.
Yeah, vomer, that's a good one. Paired or unpaired?
Unpaired. No,
Where's the vomer located?
it's located in the nose.
Right, smack dab in the center, on the midline. So just one of them.
Just one. Yeah.
What else? No, ethmoid, remember, is over here in the cranial.
Last
Oh,
one. The
palatine.
nasal.
Hold on. Yeah, nasal. And someone said palatine. I see someone wrote palatine. Real quickly, palatine, paired or unpaired?
Paired.
That
Paired.
is
Excellent. I heard
paired.
someone say nasal, paired or unpaired?
Unpaired. Paired.
Paired.
It's a paired bone.
So 2, 4, 6, 8, 10. I need 4 more bones.
The lacrimal or lacrimal.
Lacrimal. Excellent. Paired or unpaired?
Unpaired.
Paired. Excellent. Which means I only need 2 more bones.
Inferior turbinal.
Inferior nasal concha. And again, remember, I don't care how you pronounce them as long as you spell them correctly. And there are indeed 2 of those as well. Again, remember, the facial form your face, and your face has symmetry. So most of them are paired bones. You have a left and a right. The only exceptions are the vomer, which, as we talked about, is smack dab in the middle of your nose, right on the midline, and the mandible. What else is special about the mandible?
It moves.
Yeah,
It's not connected to the rest of it.
Well, it is connected, right? It is connected, although I can disconnect it sometimes to eat a really big sandwich, but it is the only one that is able to move, right? You think my lectures are bad now? Just imagine what they would be like if my mandible didn't move, All right, it wouldn't be fun to have to listen to my lecture this way. So luckily it does move. All right. There's one other thing I said at the very end, and I think some of the groups has already left, and hopefully the groups that are presented today got this. For all of these bones, you are responsible for identifying these bones. You are responsible, if it's a paired bone, for being able to distinguish left and right. So again, let me write this down. Must be able to identify all bones. If paired, you have to be able to tell left versus right. However, for most bones, only responsible for it in an intact skull. So I'm not going to have the bone chip that is a nasal bone, just a picture of that nasal bone by itself. And not only you're going to have to figure out that that's the nasal bone, but also tell whether it's left or right. However, there are some bones you will need to know disarticulated. Hopefully the groups that are presenting them today has pictures of the bones by themselves, but if not, that's okay. I have pictures of them that I can share. And so those bones that we need to know disarticulated, well, we can start easy. We already know the mandible swings freely that way. So that's going to be one of them. But it's also going to be the temporal bone, the sphenoid bone, the ethmoid bone, and the maxillary bone. These 5 bones you are also going to be responsible for disarticulated. So I could have a picture of them just sitting on the counter by themselves, and you will be responsible for them. All right. So, like I said, if you presenting one of those bones today and you don't have a disarticulated picture, that's okay. I have some that I will share with you. But the groups that are presenting today, now what we'll do is I will break you guys in your groups. It's going to take me a few minutes because I have to manually assign the groups and then put you guys into them. So you'll have that chance to do that. We'll go ahead and take our break. People can prepare. And when we come back, we will do the group presentations. Now, as I said, I want you to be respectful of the people that are doing this. When you're doing this, you do need to be on camera, but you're not being graded on your presentation skills. So again, if you don't want to look at the camera the whole time, if you want to have a piece of paper with your script on it and you want to read it off that the whole time, that is fine. Heck, if you want to hold it in front of your face while you do it, I guess that's okay as well, although you can't really see it. Like right now, nobody's looking at me, right? Everybody's looking at the middle of the screen where the drawing and where the picture and stuff is going to be. I think I've got it set up, although I think I messed up that last time. So let's do that. Hold on. No, I don't want to do that. All right, everybody, if you haven't been able to already, everybody should be able to share now. So when you're in your groups to be able to share that, perfect. Yep, that worked. So we'll take our break. The groups that are going to present today can start to get organized and can talk about what they're going to do. And then we come back from the break, we'll go into our groups. I am not going to record the groups because I know not everybody is as comfortable as I am talking in front of people. So again, this is being done for your guys' benefit. So please be attentive, please pay attention, but I'm not going to record this because, again, like I said, not everybody's comfortable being recorded and I don't want to post somebody on YouTube who doesn't want to be on there. All right. Plus Laura, she told me she's in witness protection program and so I can't post pictures of her on YouTube as well. So that's the other issue. All right. Questions on any of that?
When you break us out, do the groups that got broken out have to go back in on Canvas and log back on?
No. So what'll happen is I'll set up the break rooms and then I'll move you there and then you should be able to go there. Then at the end of the break, I will close the rooms and everybody will come back to the main room.
Okay.
Okay. So that's what we'll do. So I'll need 2 minutes, 3 minutes to get that set up. We'll take a 15-minute break. Let's call it, we'll call it 2:50. We'll get back at 2:50. That should give us enough time to get through this with a bumpy start. It's always going to be a bumpy start when we're trying to do this online, but that gives us a little over an hour and a half to make sure we get through all the material that we need to do, which we should be able to do. All right, I'll see you guys back here at 2:50. As soon as I get the group set up, I will send you to your groups. And then, like I said, at the end of the break, you won't have to do anything. I'll close the rooms and you guys will come back into here. All right, any questions? All right, I'll see you guys in 15 minutes.